we've talked about the first two tunics. The last tunic I kind of wanted to save for kind of being its own little show, in that this is a little bit more involved in terms of what's going on here. So that nervous tunic, the innermost layer is going to be what is known as the retina. So has a slightly pigmented layer behind the innermost portion of this, but then has an inner neural layer. The pigmented layer is kind of internal to the choroid and definitely attached to it, but just above that, if we're going towards the inside of the eye, you're going to be seeing the actual light detecting cells, the photoreceptors. We're going to see these are going to respond to these light rays passing through that and getting absorbed and bringing about changes in the eye. This interneural layer, again, is going to have the different photoreceptors, which we're going to see a little bit later on, are referred to as rods and cones. These have different pigments in them that absorb these light rays and convert that absorbed light rays to actual nerve signals that are going to be sent to the brain. And then we are going to be able to kind of see what's going on here with our our optical region, which is in the occipital lobe. So the retina is covering the back part of the eye. This ora serrata is about where the cutoff is. There's going to be retina back behind that, but nothing in front of that. Again, because light has to pass through here to get to the back, so there's no way for light generally to be hitting the front part of the eye behind the lens. Like I said, that ora serrata is that layer where there's photosensitive cells and non-photosensitive cells. The anterior region of this is where the ciliary body and the back side of the iris is. We're going to see the retina has a number of different layers to it, really three main layers to it. We have the photoreceptor cells, which are the rods and cones. These are the ones that actually have the pigments that react to light, and we're going to see it's a little weird in terms of how this works. You then have a middle layer here above this, and you'll see these if you look at a side of this. There's kind of three layers of nuclei. The outermost layer is that photoreceptor layer. The middle layer, which is almost as thick, is called the bipolar layer. This is what those photoreceptors connect up to and have some convergence to these ones, so multiple photoreceptors coming to a single bipolar cell. These bipolar cells are then going to be going to the next layer, and this kind of shows you these layers right here. So this would be the photoreceptor layer right here. That is a bipolar layer right here. And this third thinner layer is what we refer to as the ganglion layer. But that is what's going on with the retina in there. And it's, again, kind of an interesting one here. So you can see a little bit better here, all the pigmented layers down here. You can see the rods and cones are right here. All the nuclei of those are just above that. And then we can start looking, so that's all the nuclei of them right there that are lit up. When we then see the bipolar cell layer, that's going to be right here. And then, like I said, that ganglion cell layer is that third layer of nuclei above that. So that third layer, again, is these ganglionic cells. These are going to have convergence coming from those bipolar cells, and it's going to be these cells that the bipolar cells are connecting to determine whether or not we send a message out of the eye to the actual occipital lobe for us to say that we are perceiving light. There is some other cells that are kind of in between there that connect between some of these cells called horizontal cells. It's integrate stimuli from photoreceptors of other layers. There's also some cells called amacrine cells that help do a little bit of processing of this information between bipolar and ganglionic cells. But it's really only going to be these ganglion cells that determine whether or not they produce an action potential. Everything else is doing graded potentials. And it's going to be these ganglion cells that are then going to send the message back to the brain. So you can kind of see all the layers right here, the photoreceptor layer, and then you can see mainly the bipolar cells here, but you have some of these horizontal cells and amacrine cells in here. Then the ganglion cells are the ones that are going to be all the little axons of those coming together to make the optic nerve. There is one spot in the retina, and again, it's because if you're looking at this one right here, light is passing through in this direction, which means all these nerves are going through here, and light has to pass through those to get to the photoreceptors. But in order for all these nerves to get out of the eye, we are going to have a spot where there is no photoreceptors. That is referred to as the optic disc or blind spot. This is an area where all those ganglion axons all merge together to become the optic nerve. The other thing we have is we have a certain area where we have lots and lots of cones to give us a much sharper vision. It's kind of a yellowish region on this. It's just lateral of the optic disc and is called the macula lutea. But this fovea centralis is this little pit. It's a little bit deeper. And this is where we have almost entirely cones and very few rods. And there's very little convergence in terms of these signals, which means this gives you an area of very sharp vision, unlike our peripheral vision where there's a lot of convergence happening. So might be lots of photoreceptors talking to one bipolar cell. So you might have 400 talking to one where it's almost 1-to-1 in this fovea centralis in terms of a cone to an upstream bipolar cell. So this is showing you that optic disc right here, and you can see the blood vessels all merge to that spot too, because again, all the blood vessels have to be able to get out of the eye there. Like I said, the fovea centralis is a little bit lateral to that, and that is where you have that high density of cones in that particular area. The rest of the retina is going to be mainly rods. I mean, we do have some cones intermixed in a lot of the back part of the eye, but when you start getting the very peripheral, it's going to be mainly rods. And again, these are really good in low light and detecting movement, but not particularly good at doing sharp vision, which is part of the reason if you get something on your peripheral vision, a lot of times it's difficult for you to see exactly what's going on. You can see something's over there, but in order to know what's going on, you actually turn your head to look at it. But again, you can see the light has to pass through all this to get into these photoreceptor cells, which means the signal has to work its way back up and then out of the eye through that optic nerve. And again, the blood vessels are there as well. So again, just remind you of all the different layers we've talked about, the fibrous tunic, the vascular tunic, and the retina. You can see these different layers in there. What we're going to be doing on the last little bit here as we get to the end of this show and into the next one is we're going to start talking a little bit more about how does the retina actually do its signaling. So what is some of the physiology of this? Before we get to that, there's a few more parts of the eye we do need to talk about. We do have the lens. It is transparent again, surrounded by an elastic capsule. But the main thing here, a bunch of these cells that have lost all their organelles filled with what's called crystalline. Again, this is transparent, and the shape of this lens, much like a magnifying glass or anything else like that, is going to determine how much it bends these light rays. And that, along with the cornea, are what are going to help focus this image onto the retina, so the light that's hitting the retina is in focus and not out of focus. So you can see the lens right here, and it's again these ciliary muscles out here attached to this lens by the suspensory ligaments that allow us to do what is called accommodation. So if something moves towards you and away from you, it can actually deal with that by changing the shape of the lens so we can get more depth of field and again, more stuff in focus. Again, those suspensory ligaments change the shape of the lens. When the ciliary muscles are relaxed, there is more tension on this. This tends to pull on that lens and flatten it out. This is necessary to view things that are further away, kind of more the default position for the lens. As you move something towards somebody here, these muscles are going to relax and let this lens become more spherical. And this again is called that accommodation reflex. Their process is called accommodation, but it's the ability to track something as it gets towards closer and closer to you as accommodation. But you can see how the lens needs to change shape to deal with and focus light depending on how far the light rays are coming from. So the other thing to talk about are the cavities of the eye. So again, in front of the lens, we have what is called the anterior cavity. That can be subdivided into two separate chambers, the anterior chamber in front of the iris and the posterior chamber behind it. The whole thing on this one is filled with a substance that is a blood filtrate made by that ciliary process, which is housed close to the ciliary muscle. This fluid is very similar to cerebrospinal fluid and is called the aqueous humor. We generate some of this each day. It provides a lot of nutrients, especially the inner part of the cornea. We make more than is present in there, so it does drain out of, to me, one of the funniest names of anatomy structures. It drains out of the canal of Schlemm. So kind of an interesting one on that one. If that canal of Schlemm gets blocked, that's what can lead to glaucoma, which is a buildup of pressure in the eye because you're putting more fluid in than you can drain. So you can see this anterior chamber, and that aqueous humor is part of what's helping keeping that cornea in that convex shape, which is going to help focus the light onto the lens. And again, it helps maintain that shape, drains through the canal of Schlemm, which is a little opening here. As I was already saying, damage to this or something changing with it that blocks this a lot of times can lead to glaucoma. The main thing with glaucoma is we've talked about the eye is surrounded by this fibrous tissue everywhere, but there is one spot where it isn't, which is at that junction where the optic nerve leaves the eyeball. That is the weak point in the eye. So if you get a buildup of pressure in the eye, what it's going to do is it's going to put all that pressure on the optic nerve and then can cause damage to the optic nerve, and that can lead to blindness. What's kind of interesting is the active ingredient in marijuana, THC, tends to actually open up the canal of Schlemm, and that is part of the reason that medicinal marijuana, one of the first things it was for, was actually for treatment of glaucoma, which is just kind of an interesting aside on that one. But that is how that drains. The posterior cavity of the eye, it is made early on and it's not replaced. A lot more thickness to it. It's much more viscous. A lot of proteoglycans in there. It's kind of looks like jello-y snot. It's kind of a clear jello-y snot. And again, it's made there. It helps maintain the shape, helps keep the retina held back on there. Because one of the things that it's kind of hard to believe is that the retina is not really held to the eyeball, the back part of the eyeball, by any type of ligaments or anything else like that. It is just pressed against this, and it's really a lot of this vitreous humor or vitreous body that helps to keep that pressed back and hold the retina in place. So that kind of gives us the rest of our eye structure. So the last thing we're going to be doing on this one is we're going to talk about how do these photoreceptors work and how do they end up sending their signal back to the brain. So that'll be our last show. So I'll see you then.